Hi there, welcome to this video. So what I'm going to do here is I'm going to talk through these PAG 4 related exam questions. So they're not exactly about the experiment, but they're related to the experiment. Depending when you're watching this video, you might have not have learned everything you need to answer these questions yet, but I can talk you through it and it will help if you're watching it at the end of the course, then you will have done everything. So let's have a look at question number 1. So number 1, the diagram shows a serial dilution. So first of all, a serial dilution. This is a skill that we'll gradually develop over the course. Basically what it means is that you start with a stock solution of something. So in this case, we're starting with a sucrose solution that's 2% sucrose. So that means that in there, if, for example, it was 100 centimetres cubed, we tend to use the unit centimetre cubed, which is the same as millilitres, but it's considered more scientific to say centimetre cubed instead of millilitres. So if that total was 100 centimetres cubed, if it was 2%, then that means that they may have used 2 centimetres cubed of sucrose and 98 centimetre cubed of water. Okay, so that's how they made that. Now, the next one, they're going to kind of add these in the series, which is why it's called a serial dilution. So, what they do is they take 5 centimetres cubed out of that sample and they put it into P. Okay, and then they add 5 centimetre cubed of water. So, in here, we've got half 2% solution. And we've got half, because we've got 5 centimetre cubed of this one and 5 centimetre cubed of this one. So we've got half of this and half of this and half water. Okay, so half 2% and half water is going to dilute that down. It's basically going to half it. So this one here is going to have a 1% sucrose solution in it now because we've diluted it by half. And then the next one, they've put 5% in and still 5% water. So it's going to half it again. So it's going to go to 0.5, half it again, it'll go 0.25, half it again, 0.125. So it just halves it every time because they're adding equal amounts of this and this each time. And because it's adding from this tube to this one, to this one, to this one, that's why it's called a serial dilution. They're not getting it from this pot every time. They're just getting it from the tube before. So 1% could have been there or there, 0.5 could have been there or there, 0.25 had to be that one, 0.125 had to be that one. So your correct answer was C. And as we go through the course, we do quite a few serial dilutions. If you were adding, for example, 10 of that and 5 of that, then it wouldn't half every time. Okay, it's just because they're equal amounts on this one, it's halving. Question number 2. A teacher was reviewing some common mistakes on a variety of students' answers to biological questions. Some of the answers given by the students are written below. When investigating the effect of temperature on enzyme activity, it's important to keep the amount of substrate the same. Suggest a more suitable word than amount. Now, this is really interesting because. Sorry, it's my son. It's really important because if they're asking you to answer a question and you use the word amount, if you wrote that as an answer, they probably wouldn't give it to you. They wouldn't give you the mark. So it's nice that they're teaching you that amount is not a suitable word to use in biology. We do not use the word amount. So in this case here, we were adding substrate. We're talking about the amount of substrate. So how do we describe the amount of substrate? Okay, now in this effect here, what we're talking about is the concentration. So they're after the word concentration for this specific example. What's the concentration of substrate? Now, you wouldn't get a mark here for saying volume or mass, although for other questions, they are suitable alternatives to amount, but for substrates, then we usually talk about the concentration because we want to know relative how much concentrate is there, how much substrate is there, sorry. Question number 3. A respiration is an important metabolic process that takes place in all living cells. The black widow spider and then it gives you the Latin name, paralyzes and kills its prey with venom. The venom causes a toxin known as lactotoxin, if a human is bitten, this toxin can cause serious harm by damaging heart tissue. It causes an influx of calcium ions, which disrupts normal cell function, including respiration. An investigation was carried out into the effect of lactotoxin on aerobic respiration in humans. The rate of activity of malate dehydrogenase enzyme, a mitochondrial enzyme involved in aerobic respiration, was analysed at different substrate concentrations. Can you see there? They're talking about substrate and they call it the concentration. So they didn't say different substrate amounts, they said concentration. Substrate concentrations were used within the normal range for a living cell. So the next part wanted you to draw the graph. So it said the data shown in table 19.1 use the space provided to plot a suitable graph. Right, so we've got a result for 0. So we need on this corner here to have 0. So we'll put 0 just there. Now the first rule of graph drawing is that you have to use, oh, I don't know why it won't let me draw there. So just, oh, is it too close? Oh. There you go. I'll put an arrow there. So we need 0 on that corner, and the first rule is that your graph has to use at least half of the graph paper. Okay, so just be careful when you're thinking about your scales. So whatever goes in the first column in a table is always independent, and whatever goes in the second column is always the dependent. Whatever goes on the bottom of your graph on the x-axis is the independent, and up the side on the y-axis is the dependent. So first column always goes to the x-axis on the bottom of a graph. Second column, dependent varies, always goes up the side of the y-axis of your graph. So that's y, and that's x. So the concentration goes from 0 to 18. So on here, if we count the large squares, see if we've got 18. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So they've done that on purpose, okay, so that it doesn't fit on the axes to make you think about scale. So it basically makes it a little bit harder. So the easiest thing to do is to halve it. So if you were to do go up in twos, 2, 4, 6, 8, 10, 12, 14, 16, 18, it takes you to just here. And then you need to think, have I used half of the paper? So yes, you have gone more than halfway across, so that would be okay. So we could go 2, 4, 6, 8, draw the little lines down, 10, 12, 14, 16, 18. Okay, make sure they're exact on those. Then up the side on the y-axis, we need to plot this. So we've got 0, so we must start with 0. And our highest result looking down is 99. So let's see if we could go up in tens. So possibly 10, 20, 30, 40, 50, 60, 70, 80, 90, 100. So that works. So fill that in. Double check, does it use at least half of the paper that they've given us? And are we going up in even amounts, even increments? Yes, we are. Next thing is to label your axes. So you can copy the labels for the axes exactly from the table. So here we're going to have concentration of malate and your units, millimoles per decimeter cubed. Right, here the unit is decimeter to the -3. You might not have come across that before. So the unit could be millimoles per decimeter cubed. Another way to write exactly the same thing would be millimoles decimeter to the -3. That's just another way of writing per decimeter cubed, but you put a little minus there. So this unit up here, we could have written that millimole per decimeter cubed per second. But an alternative way is millimole decimeter to the -3, second to the -1. But that just is the same as per decimeter cube per second. Okay, just another way of writing the units. So make sure you get all your units written in. I missed the minus there while I was talking, apologies. And then up the side, don't try and simplify, just write it as it is. Reaction of malate, oops, ignore that for now, dehydrogenase, millimole, decimeter, -3, second, -1. Okay, and then you need to plot it. So you need to be really careful when you plot graphs because they're going to plot how, they're going to mark you on how carefully you can plot them. So the first one was 0, 0. Let's change colour. So it's a little bit clearer. So the first one was 0, 0. Please plot with X's, not with little circles, not with little dots, not with blobs. Please plot with X's because it's easier for the examiner to see exactly where you mean the plot to be, where the middle of the X is. So then at 1, it was 37.7. So 1 would have been in there. 37, count your squares. 37.7. You'll be able to see yours put clearer than I can. 2, 55.2. So 3, 66. 4, 74.8. Be careful, then it jumps to 6, 83.1. 8, 88.9. 10, 92.3. 14, 96.9. So it jumps again over to 14, 96.9 and 18, 99. Okay, so we plot our points like that using neat little X's. Then the last thing you need to do is a line of best fit. Now, sometimes lines of best fit in biology are straight lines and sometimes they're curved, and you need to be able to make the judgment of what's appropriate for each graph. Now, it's clear here that this graph is not a straight line. It's very clear that it's a curve. Okay. Now, a few rules on your line of best fit. You start it from a point at one end, and you do a single smooth line through as many points as you can, and you stop at the first point. You do not extrapolate, so you don't continue it along, even if you think that's obvious that that's what would happen. You do not extrapolate out either end. You only go from the first point to the last point with a single line. Equally, you don't do it sketch, so you don't do it like this. Okay, you won't get the mark if you do a sketch line. It has to be a single smooth line through as many points as possible. Now, if you can't go through all the points, try the best you can. If you put one on the left-hand side of your line of best fit, try and put one on the right hand of the line of best fit, so you're at least going through the middle of the points where you can. All right, and that'd be a super graph, be a perfect graph. Part 2 says calculate the mean increase. So mean is obviously a type of average. Mean increase in malate dehydrogenase for every 1 millimole per decimeter cubed increase in malate concentration from 1 to 10. Okay, so let's have a look at our table. So at 1, it was 37.7, and at 10, it was 92.3. I would always annotate my exam paper like that, circling the numbers I need. So the actual increase, the total increase, it says in malate dehydrogenase activity, okay, and that is what this heading is. Yeah, so that's definitely the number we need. So the first thing you need to do is find out how much it increased between those points. So you'll do 92.3 minus 37.7, which you can work out. And then, because it says for every 1 millimole per decimeter cubed in malate, that is actually an increase from 1 to 10, isn't it? So that's an increase of 9, but this wants it for every 1. So if this is for an increase of 9 and we want it for an increase of 1, we need to then divide our answer by 9 to get our final answer, which should be 6.1. Okay, so 92.3 minus 37.7 to find the total difference, and then divide it by 9 because that was an increase from 1 to 10 was a difference of 9, and they wanted it for every 1 millimole per decimeter cubed increase. All right, so our answer is 6.1, and then we need our units. So it's just the units that they used for their second column. So millimole, decimeter, -3, second, -1. You could have put the units, as we said before, millimole per decimeter cubed per second, okay? But I don't know why you would have changed it. You just probably copy it across. Now, the last thing it says in the question, always reread the question after you've done it. Give your answer to 2 significant figures. So double check your answer is to 2 significant figures. So if you've copied it off the calculator, double check that you have changed it to 6.1 so that it's correctly done to 2 significant figures. Right, next question. Lipase is an enzyme that catalyzes the breakdown of lipids. An investigation was carried out to see the effect of temperature on the activity of a lipase. Remember, lipase tells us it's an enzyme. Told us in the question as well. 5 centimeter cubed of an alkaline solution of lipid was poured into a test tube. The test tube was placed in a water bath maintained at 20 degrees C and left to equilibrate. So it left it so it got up. The contents of the test tube also got to 20 degrees. A few drops of indicator were added to the wells of the white spotting tile. The indicator is pink above values of 8.3 and turns colourless at pH values below 8.3. So it goes from pink to colourless. Once a lipid solution is equilibrated, 1 centimetre cubed of 0.5% lipase solution at the same temperature was added to the test tube. For 5 minutes at 30-second intervals, the solution was stirred. A few drops were removed from the test tube, placed in a well on a white spotting tile. The time was recorded when the solution and indicator did not remain pink. Right, so sometimes I find these all a bit overwhelming of so much information. So I like to draw an image to make it clearer. So you're welcome to do this in your exams. So it's in a water bath. The water bath is at 20 degrees C. In the test tube, we've got 5 centimeters cubed of an alkaline solution of lipid. They've added an indicator to a tile. So they've got one of these dropping tiles with the little circle dips in it, little wells in it. So they've put the indicator in there in each one of those wells. And we know that above 8.3, it's pink. And then below pH 8.3, it goes colourless. And then what did they say? For 5 minutes, once it had equilibrated, so once it got to temperature, they added into there the lipase. So they add the enzyme, and then every 30 seconds they put something that's got lipid and lipase into there. So obviously, as soon as they add the lipase into there, it's going to start to break down the lipid, isn't it? So when they put it in straight away, it will still be whole lipid molecules, and gradually the lipase will break down the lipid, and gradually the alkaline lipid will get broken down, and I guess it won't be alkaline anymore. So high pHs are alkaline, aren't they? We know our pH scale, 1 up to 7 up to 14. So this is our acid end, this is our alkaline end. So it's starting alkaline, and as it gets broken down, it's going to go down, and I guess it's going to go past 8.3. So it's going to start off pink, 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 and suddenly it's going to go colourless. So they're going to time how long does it take until it's not pink anymore, till it goes colourless. They then repeated it 4 more times at 20 degrees C, and then again at a further 6 temperatures. So they repeat 1, 2, 3, 4, 5 at 10T, then repeated 1, 2, 3, 4, 5 times at 25, etc. Why is pH not a controlled variable in this investigation? So this is interesting because usually for what we call a valid investigation, you only change one variable. So if we were changing one variable and we know that our variable that we're changing, our independent, is temperature, because we can see it's in the first column of our table, surely everything else should be kept the same and we should keep the pH the same. Well, in this investigation, we can't because pH is our independent variable, isn't it? When it said it doesn't remain pink, that means that it's changed pH. It's our dependent variable. So all you needed to say there is because it's the dependent variable. And if you weren't sure, you can tell from the table because the first heading here is always independent and the second heading across here is always independent. However many columns the independent split into, it's still all the independent variable. Next part. Part 2, identify one variable that's been controlled in this procedure. So controlled means keeping something the same. So we know that the independence temperature, so that's not controlled. We know that we're measuring pH, so that's not control. But what is kept the same? So you can read through your method. So they used the same volume of the alkali lipid. Never say amount, remember, saying the same volume of alkaline lipid. They use this, you could say, the same concentration of the lipase enzyme. You could say they use the same volume of the lipase solution. You could say that for each test, they kept the temperature the same for the 5 repeats. You could say that they kept the same time intervals between testing. So they always tested every 30 seconds. Okay, so there's lots of things that they controlled, lots of things that they kept the same that you could have said. The next one, part 3, identify one variable other than pH that's not being controlled. So reading through their method, what did they not mention controlling? So they didn't mention the concentration of the alkaline solution, the alkaline lipid. They didn't really control the volume of indicator, they just did drops, so they didn't actually measure the volume of the indicator. You could have said that, or you could have said the number of drops. They didn't mention the volume of the solution that they removed each 30 seconds. Or how many drops of sample they removed every 30 seconds. Okay, so there's a few things you could have said, you only needed one of them. Part 4, this procedure required the solution to be stirred and then drops of the solution to be placed on a white spotting tile. Let's move up. Suggest why this procedure was followed rather than simply adding indicator to the test tube, stirring and looking for a colour change. So why did they pipette it out into the dropping tile? So it's much easier to see. The reason we use these little dropping tiles a lot of the time is because it's a small volume on a white background and it just makes it easier to see. So it's easier to see the colour change. If you added the indicator to the actual reaction, because you want the reaction to carry on after you've tested it, it might affect the reaction. So we don't want that to happen. So keeping the indicator separate means it won't interfere with the reaction. So you might have said that the indicator might affect the reaction. You might have said better temperature control because you didn't have to take the test tube in and out of the water bath. I haven't got room to write that. You only needed one. But you might have written something about keeping it at a constant temperature. Part 5. What can be concluded from the results in the table about the optimum temperature for lipase activity? So optimum means the best when it's the fastest. So if it went the fastest, that means it would be the shortest amount of time. So we're looking basically for our lowest numbers. So the lowest numbers in the table, we've got a couple of 60s there, 90, 90, 60. So we've got 3 60s in that row. We've got 60, 60, 60, 90, 60. So we've got 4 60s in that row. And we've got 190 there. So they're definitely the kind of got 60s and 90s, haven't we, in around about 30 to 35 degrees C. Okay, so that indicates that that's when it's at the optimum because that's when it was taking the least time to change. Number 6, describe 2 different ways in which the procedure could be modified to obtain more accurate value for the optimum temperature of lipase activity. So the word accurate means close to the true value. So if somebody did this experiment absolutely perfectly with really top equipment, top method, what result would they get? Okay, what's the perfect answer? What's the right answer? Okay, accuracy. So how could we do the procedure differently to try and get a really accurate result? So we've come to the conclusion from ours that the optimum temperature is 30 to 35. So how could we have got a more accurate result than that? So we don't actually know if it was 70 or 80, do we, or 40 or 50, because we didn't measure those. We only measured it every 30 seconds. So you could have said that they should have used more intermediate times. What you can do also is once you know around about where it was, rather than doing them like, every 10 seconds the whole time, you could always have done it once to find out, oh, well, approximately it's going to be 30 to 35. You could have done one repeat, couldn't you, of all of them and said, well, after one repeat, we think it's going to be around 30 to 35. So let's do these ones, let's repeat it again between 30 to maybe 25 to 40, and let's go up in twos just around that area. So more intermediate times around 30 to 35 seconds, 30 to 35 degrees C. Yeah, so once we know approximately, sorry, I said times. So we could do more intermediate temperatures around those, and then we could use more intermediate times as well. Once we knew the approx times, so again, more intermediate times, because we knew the times were going to be about 60, 90. So maybe we could have taken it more regularly the whole time, or we could have done, e.g., every 10 seconds, especially between 60 to 90. Yeah, so once you know approximately what temperature and what time you're looking for, then you can try and go with smaller intervals. Now, because this was a colour change question, we could have used a colorimeter. So anything that's looking for colour changes, you can always use a colorimeter. So you might have said that as your suggestion. Okay, and then it means it's less subjective, doesn't it, if it's an actual number? Because one person watching this might have said, oh yeah, that's not pink anymore. Subjective, sorry. Might have said that's not pink anymore, whereas someone else looking might have said, actually, I think it needs another 30 seconds before it's not pink anymore. So it's a bit subjective doing colour changes. Sorry. Um, so Yeah, using the colorimeter is less subjective because you actually get a number for kind of how dark the color is or how much light it absorbs or transmits through. The other thing you could use, because you're measuring a change in pH, is you could use something called a pH meter. So a pH meter is usually just a sort of probe, like a long, thin thing with a wire coming off of it. Sometimes it goes onto a little handheld device which might have a screen on it and might give you the pH on a little screen there, or you might be able to plug it into a laptop or a computer and you get the pH on there. So pH can measure what the pH is as well. And from that, you'll get a numerical value. Um, so you'd get quantitative data. Um, the method we use kind of gives quantitative data because you get a quantity, you get a time, don't you? But it's kind of qualitative because you're recording. It's subjective. That's the thing. The subjectiveness of the color change of when it's changed from pink to colorless is a bit dodgy. Right, question number 5. A student investigated how changing pH affected the activity of pepsin. So, pepsin is an enzyme. It's an unusual enzyme because it doesn't end in A's, but it is an enzyme and it's actually a type of protease. So, it's this type of enzyme that digests protein. He used a blender to make a suspension of egg white protein in water. At the start of the investigation, the suspension was cloudy. He prepared fixed concentrations of this egg white suspension, acid, and pepsin to each of the 6 test tubes. He removed 0.1 centimetre cubed of the mixture from each test tube and used a universal indicator to measure the pH of each mixture. He incubated each test tube in a water bath at 35 degrees and timed how long it took for the egg white suspension to turn clear. He then prepared a table in which he recorded his results. Identify 3 errors the student made in the preparation of his table before he recorded his results. Okay, so look at the table. What do you think is wrong with it, basically? So if we start off here, this one here is the volume of the egg white solution. There's no units in that box. So the units are missing from that box there. You'd have written them down here, obviously. In the fourth column, he's written amount. Now, remember, we said we don't use the word amount. So you're not allowed to use the word amount. It would be better if he'd written volume because it's measured in centimetres cubed. So that's a volume. In the last column here, he's put time for suspension to clear and he's put M. So that's an incorrect unit. So the standard international unit for measuring time is seconds. So we call those SI units, standard international units. They're used across the world. And for science experiments, we should always use seconds, not minutes. Okay, that's the standard international unit that you should have used just there. So it's just a case really of just looking through the table, imagining you with a teacher. What would you mark wrong with that? Right, let's scroll down. Identify a change the student could make to his procedure that would increase the validity of the investigation. Okay, so the word validity, if something is valid, that means are we really answering the question? Are we finding out what we aim to set out to find out? And the way you do that is by only changing one variable. So that's the independent variable. And control all others. So everything else you control, you should be doing. You change the independent, you measure the dependent, and you should try and control everything else. Okay, so what did he not mention controlling that they could have controlled? So, first of all, it says he prepared fixed concentrations. He removed the mixture from the test tube using he incubated each test tube in the table. Okay, so first of all, he said he prepared fixed concentrations of egg white suspension, acid, and pepsin to each of the 6 test tubes. So, was there any measuring? How much is he adding here to each test tube? Okay, so what's the volume in each test tube? Because it needs to be the same volume, doesn't it, in each test tube? Just there. The other thing he doesn't mention is he could have used a buffer to control pH. He could have used, but main one I think really is the equal volume for each tube, keeping the volume of everything the same in each tube. I was just looking at the mark scheme while I was doing it, and it actually said less than 5% of candidates were awarded this mark. Okay, so if you didn't get that, you're not alone. But at least it's something to look for in future questions. Okay. Right, next one. State the term that best describes the purpose of tube 6. So tube 6 had 5 egg white, 2 centimeter cubed of acid, but no pepsin. So what is the point in doing the experiment without the enzyme? Well, we call that a control, don't we? If we add something that hasn't got the active thing that we're investigating, we call that a control. Another student suggested that he should repeat the investigation at least twice. How would this have improved his investigation? Now, there's a little bit of a trick to this one. Repeats increase reliability, and I always say R for repeat, R for reliability. So R equals repeat. So R for repeat and R for reliability. Okay, so wherever you get a question that says, how could you increase reliability? You say repeat. Whenever you get a question that says, why do you repeat? You say for reliability. So the answer here, we could say to increase the reliability. And then as a kind of a follow up from that, or a kind of explanation, you said you could say because you could calculate the mean of your results. So if you do 3 repeats to calculate a mean, then you should get a more reliable result. You could also say that you could assess the spread of results. Because if your results are spread, then we've got to try and work out which results are right and which ones are wrong if they're too spread out. So we probably need to do more repeats and find out actually, if you got 4, 5, 6, if we repeated it again and got 6, 6, 6, then maybe the 4 is not very good. It was So the more we repeat, the more we realise which results are right and which ones are more likely to be mistakes. Question number 6. The protease enzyme bromelain. Scroll down on my mark scheme as well. The protease enzyme bromelain can be extracted from pineapples. A student investigated the effect of changing the concentration of the enzyme and measured the time taken to break down the protein gelatin. The data from the student's experiments is shown in figure 26. Describe how the rate of reaction was calculated. Okay, so rate is basically a speed. Okay, so if you think about your speed GCSE, I don't know if you did dirty, smelly trainers, we used to do. So distance, speed, time. So if you want to work out speed, you do distance divided by time. Now, not very often in biology are we going to measure distance divided by time, but this distance doesn't have to be distance. It could be the volume per time or whatever. It can be something divided by time. So to calculate rates, we do whatever it is that we're measuring, basically, divided by time. Okay, so when you see rate, always think divide by time. That's basically how you work it out. So what is it that we're looking at? Now here, what we're going to do is 1 divided by time. Okay, so in this experiment, they measured the time taken and they don't have another measurement. So if they had a volume, then you do volume divided by time. If you've only got time, you can do 1 divided by time to turn it into a rate. And that's just something then that you just have to learn. But at least this equation, if you can think back to this, you've got the divided by time thing. And just be aware that if you're measuring time, then it's 1 divided by time. But if you're measuring volume or something, you can put volume divided by time or whatever it is that you're actually measuring. Next one, explain what standard deviation shows in table 26. So standard deviation, depending on where you are in your course, you may or may not know much about standard deviation. But basically, standard deviation is how spread out the data is. So it's the spread of data. And we could draw that in a graph. So if we had a graph like this data is not very spread. If we had a graph like this data is very spread. Yeah, so how spread is it around the mean? So the mean is down the middle. Yeah, how spread is it around the mean? How much were all the results very, very similar or were they spread out? So if we were measuring like the height of people in a class, if to get this graph, most people are the same height, a middle height, very few are short, very few are tall. If we were measuring the height of people in this class, actually there was good spread of heights. Yeah, there was quite a lot of this height, 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 quite a lot of this height. So there was much more of a spread. Okay. So explain what the standard deviation shows. First of all, you need to show the examiner you know what standard deviation means. So we can say it's the spread of data around the mean. Now, looking at the standard deviations, they're like 0.00 something. They're very, very low, aren't they? So this table shows very low standard deviations. So that means that there's not much spread around the mean. So for this experiment, it means all the repeats were very similar. And now we call that repeatability. When you repeat and they're similar, it means it's got high repeatability. Okay, now if we look for a pattern, the lowest is there. That one's a bit higher, that one's a bit higher, that one's a bit higher, that one's a bit lower. But generally, as we go down, they're getting higher, aren't they? Other than the last one. So we've got most of a pattern. 1, 2, 3, 4 out of 5 results show you that pattern. So as the concentration increased, the standard deviation increased for the first 4 concentrations. Increased, standard deviation increased. Okay, so if the standard deviation is increasing and we're getting more variety in the results, bigger spread around the mean, the results aren't as similar. That means the repeatability decreases. As you increase the concentration, the repeatability decreases because they're not as similar. Decreases. Put arrows for increase and decrease. Okay, so it might be the first time you've come across standard deviation. If so, just go with it. It's the spread of data, how spread out the results are. If we've got a low standard deviation, the results are similar. If we've got a high standard deviation, the results are spread out. That's all it means. Right, we've got another thing here that might be new to some of you. Figure 26 shows the results plotted on a graph with the standard deviations as error bars. So the mean is shown by this dot here in the middle. Okay, and then we've got these error bars, which are the standard deviations. So this one here has the largest standard deviation. You can see that's the biggest there. Yeah, this one's so tiny you can barely see it. All right, so these error bars here show us the standard deviation. So you can see that these ones, all the points were pretty much on the mean. This one, the results went down to here and up to here. This one, the results went down to here and up to here. And this one, the results down to there and up to there. Now, it's not the same as just plotting the lowest and the highest. Standard deviation is actually a calculation that we do to work out how big to draw those bars, so that we're only including the spread that's like statistically significant, really, rather than just plotting highest to lowest, because they might not really be significant. So the first one says, explain the pattern shown in the data using table 26 and figure 26. So as we were saying before, as the concentration increases, the rate of digestion increases. So we can see the points going up on the graph. We always do our conclusions from graphs. So as this, whatever it is on this axis, increases, this, whatever it is on this axis, increases or decreases or whatever. Okay, so as this increases, this increases. As this increases, this decreases, whatever. Always talk about this axis and then this one. So as the concentration, try and use all the language as well that they've used. So here they've said concentration of bromelain. Try to use the words. So try not to shorten it. So of bromelain increases, the rate, use the words of protein digestion increases. So if we were to draw a line of best fit on there, don't extrapolate past the first point or the last point, but draw a smooth curve, dot to dot-ish, if you can. You can see that it's definitely increasing, isn't it? It asked you to explain, so whatever pattern you've said, you needed to explain why it did that using the science. So it increases, which is basically describe the graph, but it's asked for explain as well. So as the concentration of bromelain increased, the rate of protein digestion increased. Now, bromelain, if you go back to the stem of the question, the bit at the start, the little intro we call the stem. So it was an enzyme, yeah, that breaks down gelatin. Okay, so it's an enzyme that breaks down gelatin. Let me just write that here so we don't forget. So it's an enzyme. So remember to annotate your exam papers as you go. That breaks down gelatin. Okay, so as we get more enzyme, the rate of reaction increases, yeah? So we can say because the enzyme concentration increases, okay, and then it starts to plateau, doesn't it? It starts to flatten off. So we can say the rate, then it'd be better to say rate of protein digestion. I'm running out of space, but the rate then plateaus. So when it starts to flatten out, always call that plateaued. So the rate then plateaus because what's happening here is that as we're adding more and more and more enzyme, the enzyme we've got too much enzyme basically. All those active sites aren't getting filled, okay, because not all active sites are filled because we haven't got enough substrate. There's the substrate is running out, which means it's the limiting factor. Remember your terminology. I'll put LF for limiting the factor. You can write it out. Don't forget, if you're asked to talk about a graph and it's got standard deviation bars, mention the standard deviations. Now, when we're using standard deviations on graphs, we look for what we call overlap. So this one here, if we draw this one in green, this one goes across at this level, so there's no overlap with these bars, but then it goes across at this level. So this top bar of this one actually overlaps with this result and this result. If I was to use a different colour for this one, you see, there's overlap there and there's overlap there. And if I use a third colour, this one, there's overlap there, but obviously not there. So these are all overlapping. These are not overlapping because the top and bottom's there, look. And that doesn't overlap with the top and bottom of that one. And it doesn't overlap with the bottom of that one either, does it? There's no overlap. These are all overlapping each other. Now, when standard deviation, when the error bars from a standard deviation overlap, that means that these results that overlap, this one, and this one, might not actually be significantly different. So it looks like there's an increase between them, but it might not be a statistically significant difference. Okay, so differences between results. So we could call those 3, 4, and 5. May be uncertain. You could say significant, statistically insignificant, or maybe uncertain, something along those lines. Okay, so when we've got standard error bars, just look, do they overlap? Is the top of this one above the bottom of this one? That's what we're looking for. Okay, next one. 7. A student wanted to investigate the effect of substrate concentration on the rate of hydrogen peroxide breakdown. So you'll have come across hydrogen peroxide. Use it in experiments from potatoes and from liver. Sorry, we use the enzyme catalase, I should say, from potato and liver, and that catalase breaks down the hydrogen peroxide, which is where will have heard of hydrogen peroxide. There are many different sources of catalase, the enzyme, A's enzyme, including ground liver or blended celery stalk. Both of these tissues could be used, but each has advantages and disadvantages. Evaluate the suitability of each of the tissues and justify which tissue is best. So evaluate. So you've got to say what you think are the benefits of liver, what you think are the benefits of celery, and then you need to make a decision which one is best and why. That's what evaluate means. So one side of the argument, other side of the argument, and it specifically said justify which tissue is best, make a decision, but don't just go with one because it said evaluate. You need to talk about both sides of it. Okay, so you could have said, for example, there's lots of things in the marks scheme. So you could have said liver contains more catalase per gram, more of the enzyme catalase per gram. You could have said the liver is easier to grind than celery would be. You could have said the celery is cheaper. So just thinking of reasonable things. It's not things you'll have learned in the specification. It will be a slower reaction, which in turn, slower reactions can give you more accurate results because you can measure it more accurately. Slower reaction, so you might get more accurate results. You could say celery's better because it's not an animal. Okay, so possible religious reasons why people think they don't want to use the liver, so it's better that it's not from an animal. You might say celery's better because it's not messy. Lots of people might not want to use liver because it's very messy to prepare and clean up afterwards. Celery could be bad, so it might be better to use liver because celery might be out of season. It might be hard to get from the shops if it's not in season, if it's not the right time of year. Celery might be a bit unpredictable because different parts of, I'm not going to write any more runner space, but different parts of the plant might have different activity levels. So, it might be a bit dependent on which bit you chose. Anyway, from that, you must choose and you must say why you chose. It doesn't matter which you choose, it doesn't matter what your reason is, but as part of answering an evaluate question, it means you always have to say one side, the other side, make a decision and say why. So you need to do that at the end of your answer. Then says the student set up the investigation using a source of catalase as shown in figure 1.1. So here's their experiment. Hopefully that looks familiar from one of your PAGs. The oxygen gas produced is collected in a 100 centimetre measuring cylinder. The gas produced was measured at 2-minute intervals. The student collected data for 2 different concentrations of hydrogen peroxide, 2 arbitrary units and 4 arbitrary units. Arbitrary units means when it's not an actual unit, it's just kind of like a comparison to one another. At the start of each test, 5 centimetre cubed of air was already present in the measuring cylinder, and figure 1.2 shows the results they saw. Construct an appropriate table and enter the raw data and the mean values. So, when we're doing a table, we must have a border around the outside. We must put our independent in the first column. So, what is it that we're investigating? What is it that we're changing? So, it told us that we were changing the concentration of hydrogen peroxide, H2O2, and it told us that the units were arbitrary units. Okay, so that's our independent. Then our dependent, what are we going to measure? We're going to measure the volume of, you could say water displaced or oxygen produced, it doesn't matter for this one. That's fine, and you need the units there, so that would be measured in centimetre cubed. Okay, did it say they were going to repeat or not? Do we need to divide that more? Oh yeah, because you can see here they've got 3 repeats for 2 and 3 repeats for 4, but they're just all muddled up. So we can see they've done it 3 times. So we would put that into the table. We'd put repeat 1, repeat 2, repeat 3, and the mean. So we divide the dependent then into the 3. Sorry, those lines should line up. Not very good, is it? We should also use a ruler. It's hard to fit it all in. Let me try and undo that. Let's try and just do that a bit neater, shall we? I only left space for the heading, so let's make it. So we've got space for everything. Let's start again. So, first of all, concentration of hydrogen peroxide, which was measured in arbitrary units, they said. And then our dependent variable was going to be the volume of water displaced, or you could have said oxygen made, depending on how you're going to read those tubes. And that will be centimetres cubed. Okay, then we've got repeat 1, 2, 3, mean. So please make sure you've used a pencil and a ruler. Sorry, I'm not very good on here. There we go. That's where all your lines belong. So you should just have one section here for independent, one section here for dependent, and dependent can then be subdivided as needed. So we only had 2 and 4 in there. So make sure you got your box drawn around that. Join all these up, please. Do it neatly. Okay, so for 2, we've got this one here. So then it's a case of reading the values off. So for 2, if the water started, what did it say? It started at 5, did it say? 5% of air was already present in the measuring cylinder, and they were 100 centimetre cube measuring cylinders. So these measuring cylinders aren't the kind that have the scale upside down. So you've got to imagine that this goes up, and it started at 100, yeah, and then it started with already 5 of air, so it started at 95. So if it started at 95 and it went down to here, so that looks like it's got 10 little lines in between. So 40, 41, 42, 43. That looks like it's on 44, doesn't it? So what's the difference between 44 and 95 is basically what you need to work out. So that's 51. So each time you need to work out the difference between where the level is at now and 95. So the first one I'd say is at 51. So the next one I'd say is at 29. So 29, 30, 40, 50. So you need to do 95 minus 29. Then the next one you need to do 95 minus, what would you say that is? My computer screen's so small. Hopefully you can see them better. 43. I've just realised that one belonged down here, didn't it? That was a 4. That one was a 4, wasn't it? So these are the 4 ones, and these are the 2 ones. So this 2 1 is at 38. So you do 95 minus 38 for that one. For this one, I'd say that's on 34. So you do 95 minus 34. And this one I'd say is on, or this 41, sorry, down here is on 36, is it? So 95 minus 36. So you needed to work those out. Don't write those calculations in, write the actual answers that you got. And then calculate the mean. So add the 3 together, divide by 3. Okay, so it asked you to put the raw data in, okay, and the means. So making sure you've done that. Part C, catalase activity can vary between different tissues within the same organism. A second student carried out an investigation using catalase from muscle tissues. All the samples were taken from the same individual organism. The results are shown in the table below. Complete table 1.1 by calculating the variance for each set of data. Okay, so variance is S squared, so it's standard deviation squared. All right, so even if you haven't got onto your statistics part of the course yet, you can see that standard deviation is S and variance is S squared. So it's just the value for standard deviation and square it. So we should have here the leg muscle 29.16 and the wing muscle 136.89. Right, this bit looks a bit daunting, but again, it's just some statistics. You'll get taught how to do it. If you want to have a go now, that's fine. If not, you can wait, doesn't matter, but it's nice to just come across it. So when we do get to it, it's not going to be quite as daunting. So it then says the student decided to carry out a t-test to determine the two sets of data were significantly different from each other. Calculate the t-value for the data for table 1.1. Use the formula shown below. So for these statistics, you don't have to learn the formula. You just have to be able to use the formula. The correct answer is 3.558. Okay, I'll tell you a little bit about these. So then it's not as daunting as it might look. Okay, so first of all, x bar is the mean, so you can see that the value x bar 1, x bar for the set of data 1 is just there. Okay, then x bar 2, so that means x bar means x with a line on it, mean for data set 2 is just there. Then you're going to divide it by the square root of, and then it asks for S, which is standard deviation. Look, so S1 squared, so that's your S1, 5.4, and square it. And then it asks for S2 squared. So here's your S2, square that, pop that in there. Then N1 is just the number of samples. So in sample set 1, we've got 10, and in sample 2, we've got 10. So number in set 1 is 10, number in 2 is 10. And you just pop all of those numbers into this big equation, okay? And then you can just work it out little bit by bit, and eventually you should get that answer. Now, we'll teach you to do that in class, but if you've just come across it a few times in these questions, it will be definitely much less daunting when we get there. Part 3, the student stated that there's no significant difference in the time it took to collect 2 centimetres cubed of oxygen, so the activity of the catalase in both types of tissue is the same. Use table 1.2 to decide if the student is correct. Explain your answer. Okay, so here we've got something called degrees of freedom and then levels of probability. So 0.05 is basically 5%, 0.01 is like 1%, 0.001 will be like 0.1%. Now, this is just, again, following up the statistics. Okay, you'll learn how to do this as we go through, but it's nice to see it in advance and it makes it easier when we get there. So, for biology, we tend to use the 5% column. So we're going to be looking at the numbers in this column here. So one of the things you need to know is to look at the 5% column. Now, using your answer from the previous part there, we had 3.558. Now, We have to know how to work out degrees of freedom, which is obviously just something you need to learn how to do. But basically, it's the total number of here, it's the total number of readings minus 2, so it's 18. So our critical value here that we're interested in is in 2.101. So looking at our results down here, our result was 3.558, and we're interested to say, is it bigger or smaller than the critical value? So 3.558 is bigger than the critical value because it's bigger than 2.101. Okay. At the 1% significant, at the 5% probability level. Okay. And therefore, what we would say is that we reject something called the null hypothesis. Okay, which means that the difference, what they're actually asking you in the question, the difference in the enzyme activity is not due to random chance. So a null hypothesis is saying actually this is just caused by random chance. So, oops, sorry, chance with a C. If we're rejecting the null hypothesis, then we're saying it's not due to random chance. It's actually due to the biology of what's happening, what we're investigating. All right, so if you haven't come across that yet, it might seem really tricky, but the more you can just listen to my videos and get an awareness of how to do the stats, the easier it will be when we get there. Part 4, comment on the precision of the data obtained for the two types of muscle. Okay, so precision this time. So precision means basically how close together the results are. Okay, so how close together are our results? So we look in our table. Okay, so when they took 10 samples, how close together are our results there? How close together are our results there? So if we were to just look at the range from highest to lowest, on the leg, if we look for the lowest number, and we look for the highest number, whereas on here, if we look for the lowest, and then we look for the highest, this one has got more of a range, hasn't it? This one's got less of a range. So we'd say that the leg muscle the leg muscle is more precise because we've got a smaller range because our results are closer together. Okay, so precision is how close together our results are. Okay, so the leg muscle has greater precision okay, because the results are closer together. All right. We can also tell a little bit about precision from standard deviation because standard deviation tells us the spread around the mean, and because leg muscle also has a lower standard deviation, that means it's less spread out. So that's another piece of evidence for the leg having a greater precision. Right, we're nearly there. It's a long one, isn't it? Number 8. Chymosin is a digestive enzyme found in the stomach of young ruminants. Its role in digestion is to coagulate or clot milk by catalyzing a reaction that results in the precipitation of milk proteins. A student planned an investigation into the effect of temperature on the rate of reaction catalyzed by chymosin. Okay, so if it's the effect of temperature, that's our independent variable, and the rate is our dependent. Figure 1.1 is a page from their notebook showing the procedure used. Set up 5 water baths set to these temperatures. Get a beaker and we're going to add 10 centimetre cubed of milk and we're going to place it at 20 degrees for 5 minutes. Then we're going to get a test tube and we're going to add 1 centimetre cubed of chymosin. Now let's, can't spell it. It's told us that chymosin is the enzyme. It told us that in the question. After 5 minutes, add this into here. Start the stopwatch, observe it every 30 seconds until clots appear in the milk and record when the top clots are clearly seen. Repeat it for these temperatures using clean equipment each time. Name 2 pieces of equipment that could be used to accurately measure the volume of chymosin in the procedure. Okay, so measuring volumes. A beaker, absolutely not. It's hardly got any graduations on the side of it, and also it's very wide, so it's not going to measure very accurately. Okay. Best to say a pipette, but say what type of pipette. So we call it a graduated pipette. We could say a volumetric pipette. We could say a syringe. We could say what type. So, for example, a 1 centimeter cubed syringe. Okay, but we need to be really, really specific. All right, so we need to use equipment that's small enough for what we're measuring. Because we're measuring really small amounts, even measuring cylinders are probably going to be too big. You're not going to get the mark even for a measuring cylinder, and definitely not a beaker. We don't measure in beakers. Next one. Oh, it's going into the mark scheme. Look, we are nearly there. Explain why the range of temperatures used in the procedure was suitable for this investigation. So the temperatures that they used were from 30 up to 60, or actually from 20. The first one was at 20, wasn't it? So they did 20 to 60. So we don't always want to go 0 to 100. That's the temptation. Always say 0 to 100. It's not good. You need to use something that's kind of realistic. So it told us that it's found in young ruminants. So we know it's found in mammals. And we know that mammals work best at 37.5 degrees. Okay, so mammals, optimum temperature is 37.5 degrees. So whenever we're doing experiments to do with enzymes that are found in mammals, we need to use temperatures that are around 37.5 or 37 degrees, so which is in the middle of the range they chose. Part B. Another student was asked to repeat the investigation to obtain a set of replicate results. Suggest 2 additional pieces of information that would be needed to obtain reproducible results. Give a reason for each of your suggestions. So, what haven't they told us in the method that we would have needed to know? So, you could have said something to do with the pH. So, we need to control the pH. What pH should it be? And the reason why is because pH will affect the way enzymes work. If it's not the same, it will break bonds, it will change the 3D shape, make it work differently, reduce their activity probably. You could have said the concentration of the enzyme. They didn't mention enzyme concentration. And that's because obviously if we've got an increased concentration, the rate will be faster. Yeah, so it's going to affect the rate of reaction. They said about using milk, so you could have said something like use the same type of milk. Because they might have different concentrations of the protein, or of the the protein, the substrate that's going to get broken down. You could have said concentration of substrate, so where I put concentration of enzyme, I'll put splash substrate, because obviously if you've got more substrate, it's going to work faster. So, same reason actually, I won't write it out again. Just increase the concentration of the substrate, it will make the rate faster. Okay, so any of those, but make sure you've got reasons. Part C, let's go down a little bit. Chymosin is used in the commercial production of cheese. Chymosin acts on proteins in the milk until it clots to form the curd, which is soft cheese. An enzyme inhibitor is then used to stop the reaction catalyzed by chymosin. During the commercial production of cheese, it's important that the correct concentration of the enzyme inhibitor is used to stop the reaction. Explain why. Okay, so if they use too little, if they didn't use enough inhibitor, what would happen? the reaction might not stop. If they use too much, it might affect, I mean, if it stops, it stops, but it might affect the quality. If it's got too much inhibitor, it might affect the quality of the product. And then it's really a balance between the two, isn't it? Because you might say, oh, too much doesn't matter, because as long as there's enough to stop it. But then too much is actually going to, you've got to then think about the cost, haven't you? If you use too much, it's going to cost too much money. So you want to use as little as you can that makes it stop in time. Okay, we got one more left. Last one. Describe how the procedure used in A could be adapted to investigate the effect of concentration of enzyme inhibitor on the activity of chymosin. Okay, so this one here is a long answer, and it's not going to be a case that we just count up what you've put in order to give a mark. It's not like a point per thing that you say. But whenever we're needing to um, look at, I was just seeing if I could plug in the computer, sorry. Let's find it. Um, Whenever we're looking to design an experiment, there's a certain kind of structure that we can give our answers just to try and stop us missing anything out. So we could start off with the independent variable, the dependent variable, and the controls. So it tells us that we're investigating effect of concentration of enzyme inhibitor. So concentration of enzyme inhibitor. That's what we're investigating. That's what we're changing. Now, your independent variable, you always want 5 different concentrations. So you can say what those 5 concentrations are going to be. Dependent, what are we going to actually measure? We're going to measure the time it takes to become transparent. And then you need to think about control variables. So you could say temperature, say how you're going to do it. So you could say water bath, say what temperature it's going to be at. Well, 37 is a good one if it's going to be in a mammal. You could talk about volumes, state what volumes you're going to use. It doesn't really matter too much what they are, but state the volumes with units. Okay. So you need to list numbers here. You need to list numbers here. I've put a number there, okay? Use numbers in it as well. Then you're going to go through your method, how you're going to do your experiment. So use the similar method to what's above. And go through your method. Try to, for each step, justify why you're going to do it. When you get to the end of your method, it's going to be a good idea to always just say that you'll repeat each one 3 times. You're going to calculate a mean and rough a repeat. That's to make it more reliable. Okay. You can also say at the end, you could say that you would do some statistics to make it to see if your results are significant. Remember, if you've used anything that's hazardous, you can mention that. So it might be a good idea to put it here actually. Risk assessment. So anything that's hazardous, you can mention your risk in your risk assessment. So perhaps the enzyme or the hot water bath, something along those lines. And then you need to mention the equipment. So either you can put an equipment list or you can mention your equipment as you go through your method. It doesn't matter either way, but make sure you mention what equipment you would use for each thing and why it's a good piece of equipment. So, for example, if you're measuring volumes of like the enzyme and the substrate, make sure that you say you'll use pipettes or syringes. Make sure you use really small equipment because you're measuring small amounts. Distilled water, not just water, be specific. If you're going to do dilutions, you can explain how you're going to do them. If you're mentioning volumes, say numbers, so just copy similar numbers from their one. Make sure you use water baths. How are you going to determine the end point? So um, if it's going to go transparent, how can you tell? You could draw a cross on paper underneath or something like that, or you could put it in a colorimeter, take a sample, put it in a colorimeter. Okay, so lots and lots of ideas there, but hopefully that structure gives you an idea of how to get started on all those types of questions. And we'll leave it there.